This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 26 for March 27th, 2009. From sunny New York City, I'm Vincent Racaniello.
And from sunny Western Massachusetts, I'm Alan Dove.
Joining us from Florida is Rich Condit. Welcome to TWiV, Rich.
Thank you. It's great to be here. And I hate to disappoint you, but it's been socked in all day. It's really overcast here. It should be sunny, but it's not.
That's interesting. Last week we had Luis Villarreal, and it wasn't sunny in California. Now in Florida. And those are two of the places where it's always supposed to be sunny. Well, we're happy to have you with us, Rich.
I'm really glad to be here. I've been looking forward to this.
So Rich is a professor of molecular genetics and microbiology, University of Florida at Gainesville. Is that all correct?
That's all correct.
How long have you been there, Rich?
I have been here for almost 19 years. For 12 years before that, I was at the State University of New York in Buffalo in a biochemistry department. And that's my whole professorial run. But wild horses couldn't tear me away from this place. This is great.
You like it, huh?
Yeah, it's wonderful.
I hear you can walk to work. Is that true?
You can. Actually, I ride my bike usually. My house is about 3 miles from work, so I commute on my bicycle.
Sounds great. You know, Harold Varmus here in the city does that, I understand.
I've heard the same. That's right.
Well, Rich is an expert on poxviruses, and that's going to be our theme today on TWiV. And as usual, we have couple of interesting stories, and I've picked out a few pox virus stories to make sure you're happy, Rich.
I'm sure I'll be delighted.
Although I know you like all viruses, correct?
Well, actually, I noticed that one of your bits is on polyomaviruses, and in fact, I did a postdoc on polyomaviruses, and I do like all viruses and retain a special fondness for polyomaviruses.
I remember when you and I were on study section together, you were always fascinated by all the things you had to review.
Yeah, in particular, this dude, I remember a polyomavirus that it's a wasp virus that is just outrageous. That's a whole story in itself.
In fact, there was a paper a few weeks ago that we talked about on TWiV because the sequence of the wasp genome revealed that the virus is actually encoded in the wasp.
Right.
It's by a French group. Remember that, Alan? We talked about that.
Yeah, very cool stuff.
And I actually wanted to get you on for that, but it didn't work out. Today we'll do pox, and I'm sure you'll be excited. And polyoma. Who did you work with on polyoma?
Bob Kamen. It was a large group at the Imperial Cancer Research Fund in London that included Bob Kamen and Lionel Crawford and Alan Smith and Mike Freed and Beverly Griffin. It was a whole floor. It was a wonderful place and a wonderful time.
I know a lot of those names, yeah. Well, this week polyoma came up, and then I found a brand new paper in PLOS Pathogens, so we'll talk about that. But before we do, Alan Dove, you forgot to mention Jeremy Luban as a member of your thesis committee.
Oh my, yes, you're absolutely right.
He sent me an email because he listened to TWiV.
Yes.
So, Rich, a couple of weeks ago we had Hamish Young on TWiV.
Mm-hmm.
Alan said, "Is it the goal to reconstitute my thesis committee?" And I said, "Who else was on it?" And he said, "Well, Jim Hogel was on it and Alan Stahl." And then my former colleague, Jeremy Luban, sent us an email saying, I was on that committee too. So
yes, he was.
I think he felt left out.
He was on TWiV, so...
He was. So I had to mention that because we don't want to make our participants feel left out.
No.
Okay, so our first story came out in the news this week. It's a story that came out of Biogen. My colleague here, Saul Silverstein, said, you have to look at this Biogen story. They're using a malaria drug for virus infection. I didn't know what he was talking about. So the story is very interesting that a number of years ago, Biogen started selling a monoclonal antibody to be used for treating multiple sclerosis. Now, multiple sclerosis is one of these autoimmune diseases, and in this case, the target is myelin. You get an immune response against myelin, and you lose it, and as a result, you have neurological degeneration, essentially. And this antibody that they sell, the trade name is Tysabri. It's an antibody against this cellular protein alpha-4 integrin, which is basically a leukocyte membrane protein that's involved in leukocytes going from the blood into tissues. So this antibody inhibits that trafficking, and has been shown to be effective in treating multiple sclerosis and I think also Crohn's disease, another autoimmune disease where the target is the GI tract. Okay, so they released this drug a number of years ago, and within a short time, a number of cases of this really rare brain disease called PML, progressive multifocal leukoencephalopathy, arose in some of these patients. And it turned out that, well, this is one of these polyomaviruses that causes this disease. In this case, it's JC polyomavirus. Many people are infected with these viruses, and there's usually no consequence. But when you get immunosuppressed, they take off. And in the case of JC, it grows in oligodendrocytes in your brain, and within a short period of time, you die. So this was not a good side effect. What they did was to do a screen of FDA-approved drugs to search for one that would inhibit JC replication. And they came up with this anti-malaria drug, mefloquine.
Right.
And it turns out to inhibit JC replication. So now they're doing a clinical trial to see if you get PML in patients who are getting this Tysabri, if you can treat them with anti-malaria drug. So I thought this was an incredible confluence of all sorts of things. You have a treatment for MS, and then you have this virus infection, and that got me to look at polyomas in general. So do you know anything about PML, Rich?
It's not an uncommon end-stage disease in AIDS patients.
That's
it shows up very commonly there. I'm curious about this malaria drug. Is the mechanism of action known?
I don't believe it is.
Because, the polyomaviruses are really difficult to handle because they're so host-dependent. And so finding a stage of a portion of the virus replication cycle that is unique to the virus that you could target with some sort of drug is tough. So, for example, most of the nucleoside analogs that are replication inhibitors that you might expect that might work on some viruses, I would expect would not be effective on polyomaviruses because they're using the host apparatus almost exclusively for replication. So the mechanism of action of the malaria drug is of considerable interest in this case. That's very interesting.
There's one paper on this, which was published just in March in Antimicrobial Agents in Chemotherapy, Identification and Characterization of Mefloquine Efficacy Against JC Virus in Vitro. And this is the description of the screen where they used a collection of 2,000 approved drugs, and they pulled out mefloquine. And it doesn't have a mechanism.
That will be of significant clinical interest, not just for PML, but there's the BK virus in transplant patients. Again, another sort of immunosuppressive situation can cause hemorrhagic cystitis, and it can be very nasty, and there's no treatment for that.
Right.
And it'd be interesting to know if this same drug would be active against that virus, and of course both of them in a clinical situation.
I'm sure it's being looked at. You're absolutely right, yeah. This is very interesting because I think just last week or the week before we did a paper on TWiV where people had made pseudotypes of henipavirus glycoproteins with VSV, and they looked for inhibitors using FDA-approved libraries, and they pulled out a malaria inhibitor, which inhibits henipavirus infection. And in that case, the mechanism was inhibition of, I think it was cathepsin L, and that was required for production of the henipavirus in cells. I don't know if it's coincidental, but it's interesting that two separate screens now for antivirals are pulling out malaria compounds.
No, that's very interesting.
I don't know what that means, but so I was getting ready this morning to do TWiV, and I found a paper in PLOS Pathogens by Bob Garcia, who I know, and you must know also, Rich.
Yes, I do.
At Denver, called Epidemiology of Human Polyomaviruses. So it's perfect because in looking at the Biogen story, they mention how these polyomas are prevalent. And here, Bob actually looked at it. So he has a collection of 1,500 human sera from a blood bank, basically healthy adult blood donors. And they produced capsid protein VP1 of these various polyomas. And there are 5 so far. There's JC and BK. There's Merkel cell polyomavirus, then KIV and WUV. He looked for reactivity with VP1. And here are the numbers. They're quite interesting. BKV 82%, JCV 39%, LPV 15%, KIV 55%, and WUV 69%, and the Merkel strains 25% and 42%. So pretty high.
And these are in healthy adult blood donors.
Yeah, exactly. And then they did 721 children's sera, and they said the numbers were similar. And the suggestion, of course, is that that's when you get infected with these viruses when you're a kid, and then you keep them your whole life.
That's right. They hang around. At least BK does hang around persistent in the kidneys. I think a lot of them home to the kidneys.
Do you make virus all the time, Rich?
My understanding is that if you look hard enough, you can find a little bit of virus in the urine. I don't want to commit myself to all the time. Certainly, in immunosuppressed people, it pops up in the urine, I think, pretty routinely. But my understanding is that, it's not like a herpes virus latency where you're not making virus, that if you look hard enough, you can find a little bit of virus ticking over all the time.
Very interesting, isn't it?
One of the things that really interested me about, I looked up that paper because you previewed it, and one of the things that interested me about that was that they look for SV40 as well.
Right.
A monkey virus. And after they tried to eliminate various possibilities of cross-reactivity, came up with a number of like 2% SV40 positive. And of course, this goes back to, you may have discussed this in a previous session, the notion of people being vaccinated decades ago with the oral polio vaccine that was contaminated with SV40.
Right.
And that possibly creating some sort of problem. Apparently it has not. They did the age profile on the people who had the 2% who had something that looked like SV40, and it was apparently distributed throughout age groups, so it included a lot of people who would not have gotten that vaccine. And it seemed to me the implication in the paper was that they didn't really think that this was actually SV40. But maybe indicated that there's cross-reacting polyomaviruses out there that they just haven't identified yet. That they're
be interesting
up.
If you could do PCR on those people who tested positive for SV40 and see if you can actually get the virus out.
Absolutely.
Yeah, the nature of the blood bank, of course, being that you wouldn't be able to do that.
Right.
This is stimulating some future studies like that, I think.
Sure.
Yeah, they say that the 2% indicates does not support its role in human disease.
Right.
So that could be cross-reactivity, or it could actually be a bona fide infection. And one of the claims for this, we haven't actually talked about this contamination of SV40 before. So about a few million people, I believe, got SV40 in either the inactivated or the infectious polio vaccine.
Well, I was certainly a candidate. I don't know if I actually got it, but I was waiting in line, I'm sure. I probably got it.
Yeah, you got it in the 60s, I would guess, right? Or the
right,
50s.
Because I remember lining up in the school gymnasium and getting my sugar cube with the Sabin vaccine
yes,
on it.
I do as well. And in fact, I have my immunization card to this day, which maybe I'll post it on the website so everyone can see. I think it was 1960 or 61.
That sounds about right.
So maybe we should look for SV40 in us, Rich.
Absolutely.
It's not a hard experiment.
Well, maybe all we got to do is visit Bob.
Which is always nice to do. Bob's a nice guy. In fact, I'm thinking we should get Bob on a TWiV to talk about this because he knows a lot more than I do. So some people claim that initial inoculation of SV40 into the population, in fact, allowed virus spread. The problem with that is, of course, SV40 causes tumors in some animal, right, Rich? What kind of animal does it cause tumors in? Mice? Monkeys?
Mice, I believe. Well, no, it's a monkey virus. My spin on the polyomaviruses with respect to tumor formation is that basically they cause tumors in heterologous hosts where they can't go through a lytic infection.
Right.
Where they go through an abortive infection. So, so if you put them in the wrong species, if they can initiate an infection, there's an opportunity for the T antigen, which is the transforming gene, to integrate into the host chromosome, and that's when you come up with a tumor. And I believe that in vitro, SV40 will actually transform human cells, because I think the infection on human cells is abortive, whereas monkey cells is a productive infection. And that's where the fear comes from, that SV40 in the human population could cause a tumor. But as you've already said, there's no actual evidence for that.
Right, but there's a lot of controversy and there's a lot of litigation going on, and some people feel that It's associated with human tumors, and I know that having worked on a litigation case recently, I heard all about this, and the trials are ongoing, so we'll hear more about it most likely. So that's cool. I like that story. The next story is very quick, and that is the flu vaccines for next year have been selected. I thought this was worth mentioning. Maybe most people don't know that this happens very early on, so here it is. This committee, it's called the FDA's Vaccines and Related Biological Products Advisory Committee, met in February 2009 to select the flu strains for next season, because now they have to start being manufactured. So we have 3 components. There's a H1N1 and H3N2 and a flu B component. So the particular strains. The 2A components are from Brisbane. B is also from Brisbane, all 3 from Australia. The idea being, this is what I understand from the flu people, what's prevalent down there now eventually makes its way up north and spreads globally. And I saw some very interesting maps of that kind of spread at a recent Swiss virology meeting. And these are what are predicted to be the prevalent strains in the rest of the world next season. So we'll see. They don't always hit it right. But if you're interested, we'll put a link to the CDC site where they say how they do this. They basically collect strains all over the world and do antigenic relatedness and see if they can find some that are more related to the majority strains and so forth.
And then the vaccine companies have to race to prepare the correct vaccine. And in the case of flu mist, where they actually have to create recombinant viruses from these, it's even more of a project. It's an interesting process.
Actually, even for the egg-grown inactivated viruses, they do make a high-yielding recombinant.
Oh, okay. I wasn't aware of that.
These clinical isolates typically grow very poorly in eggs, and so they swap out the most of the genes with a high-yielding egg strain, and they keep the surface glycoproteins there. I know this because when I was a graduate student, the chair of my department was Ed Kilborn, and he used to do that in his lab. He used to make this recombinant strain. He had an army contract to do that, and I believe they still do it. But if I'm wrong, one of our listeners will correct us, which is the good thing about this new media, which we don't resent at all. Absolutely not. Okay, now we have a couple of stories right up your alley, Rich, and I'm hoping you'll just set us straight on all of these.
Well, I'll give it a rip.
The first one is a paper I found called New Cell Culture Propagated Smallpox Vaccine, a little test on a smallpox vaccine that they have grown in cell culture. So why is this of any interest at all, Rich?
Well,
or you could say it's not of any interest, too, if you
Well, no, it is of interest, at least to some people. The interest in smallpox vaccination in general has increased over the past, 8 years or so, ever since 9/11 and the anthrax incident and the resurgence or the emergence of the possibility of a bioterrorist event. Okay, and so smallpox is sort of number one on everybody's list of possible bioterror agents. And when discussions about that possibility arose, the first thing that people did was look at what our vaccination status was and what our stocks of smallpox vaccine looked like. Because, well, I suppose there's a little bit of prehistory here that's important, because used to be everybody was vaccinated against smallpox, and in 1978 the disease was declared eradicated, and at that time the World Health Organization, at the same time as they declared smallpox eradicated, they said people should no longer vaccinate against smallpox because now the risk of dying from complications of the vaccination is greater than the risk of dying from smallpox. And the risk of dying from complications of the vaccination is about 1 in a million. In fact, in most developed countries, the practice of smallpox vaccination had fallen off for a decade or so, even before 1978. In this country, in the 60s and early 70s, even though it was continued by some physicians.
The complication is the 1 in a million. It's not smallpox. It's something else, right?
No, it's. I'm glad you asked. In routine vaccination, there are basically 5 different complications possible. One is just accidental vaccination, because the vaccination consists of scratching into the skin on your arm some live virus, and you get a blistering lesion that grows out of that, and in that blistering lesion is live virus. And so if in caring for that, you can wind up with the blisters will pop and stuff. And so in the fluid that comes out, there's live virus, and you can get this on your hands and transmit it to other people. The virus likes to grow in skin and mucous membranes, and in skin it grows really where there's just fresh wounds. Okay, most skin is tough enough. Healthy skin is tough enough to resist infection by the virus, but in a fresh cut or in a mucous membrane, it'll grow. So eyelids and cuts in the skin. So accidental transmission of the virus from a fresh vaccination to someone's eye or another cut was a complication. Another complication was what is called progressive vaccinia, and this is usually in immune-compromised individuals where the infection spreads out from the initial site of the vaccination, and that can be fatal. Another complication is called generalized vaccinia, and that's where you actually establish a viremia from the vaccination site, and it looks kind of like smallpox. You get a disseminated pox-like disease. Another complication is eczema vaccinatum, I think it's called, and basically that's if a person is vaccinated who has eczema, bad idea. They of course have lesions or all cut-like lesions, if you like, all over their skin, very sensitive skin, and the vaccine the virus can spread on that, and that can be a life-threatening disease. And the last complication is post-vaccinal encephalitis, inflammation of the brain, and in fact, that's the one that is the most fatal. So the risk of dying from a vaccination in a normal population is about 1 in a million. And most of those are post-vaccinal encephalitis. Most of these things you can screen against. You don't vaccinate people who are immune compromised. You don't vaccinate people who have eczema. There is a rare complication. On rare occasions, if you vaccinate someone who's pregnant, the fetus can be infected, and that's fatal to the fetus. So you don't vaccinate pregnant individuals. And that eliminates most of the complications. There's no way to screen. There's no contraindication there's no way to screen against people who are going to get post-vaccinal encephalitis.
Right. Okay. Great.
So, how did we get into all that?
So we were talking about the vaccine.
Okay. So because of complications from vaccination and because smallpox had been eradicated, nobody vaccinated anymore. And so we got a 30-year period of time, at least from 1970, almost 25 years, from 1978 to 2001, 2002, when all of a sudden people are starting to think, maybe smallpox could be a problem again as a result of a bioterrorist event. What do we have to deal with this? So the first thing I did was to look at what stocks of vaccine we might still have around. Now, the vaccine that was used during the smallpox eradication campaign and routinely in this country was called Dryvax, produced by Wyeth. Right. And that's produced on animals, produced by you basically give a calf a confluent vaccination on their shaved belly and then harvest all the fluid from that, grind it up and spin it to sort of clarify it, add a little phenol to inactivate any bacteria that might be around, and then freeze dry it.
Old school manufacturing.
Old school. Boy, this stuff would never make it past the FDA now. And you reconstitute it and then administer it with this unique tool called a bifurcated needle, which is basically a stainless steel thing that looks kind of like a two-tined pitchfork, that the little groove holds a couple of microliters of the virus, and you just touch that off on somebody's skin, and then it's got these flared points on it that you prick through the fluid. So you draw just a little bit of blood, and that seeds freshly broken skin with the virus.
Can I ask you just quickly, don't we have stockpiles of Dryvax which were produced recently? And were they produced in this calf immunization way?
Those stockpiles of Dryvax were produced in the 50s.
Okay. We have no recently produced stuff then?
No.
Okay.
Not to my knowledge. But one of the first things they did was ask whether they could extend that stuff by diluting it. And they discovered that you could dilute it at least 5-fold and maybe even 10-fold. So there are thousands of doses of Dryvax around. And in fact, We vaccinate people in our laboratory as a safety precaution, and although I haven't checked yet, checked recently, I think they're still getting dry vax. So at any rate, there was some motivation with the biodefense stuff. There wasn't enough dry vax around to cover, the whole country in the event of a bioterrorist attack. So that stirred up motivation to address that problem, create some new vaccines. Obviously, the dry vax method, as we've already discussed, is not going to pass muster, and there are complications associated with dry vax. So the first thing that was done was to create a strain that's called acambus, which is just a plaque, something that was plaque purified out of dry vax and tested thoroughly. So at least we have a characterized strain, and that stuff was grown in cell culture, and that's been stockpiled and licensed, and that's available. Unfortunately, that has some of the same issues of side effects as does dry vax. And so more attenuated vaccines are desirable. Now, this one that you pointed out in the reference that you sent is a strain LCM18 something, I think.
LC16M8, right?
There you go, LC16M8. So that's a strain that was actually developed in Japan as an attenuated strain, as a laboratory attenuated strain, in the way that people do this kind of stuff, where you serially passage virus on cultured cells or eggs. And I forget exactly the procedure in this particular case, although it was only a week or so ago that I heard a seminar on it. And it was plaque purified at a couple of different points, and at one point they had a virus that was temperature sensitive, and it was passaged further, and et cetera. But the whole idea is if you passage these things for a long period of time in culture, they'll lose some of their virulence properties. And so the result was a vaccine that was significantly attenuated in terms of side effects, that was actually used in the Japanese population. I forget exactly when, but they have a lot of statistics on it, not only in the human population, but also in model systems that say that this, in fact, is a significantly attenuated strain, and therefore is actually much safer in humans than dry vax or perhaps the acambus strain. And this is licensed in Japan, and the Japanese are trying, and you can grow it in cell culture. Okay, it is grown in cell culture, and so the Japanese are trying to get licensure in the U.S., and so that's a reasonable idea. There's one other thing on the table, and that is another attenuated strain that was developed in Germany called MVA. And that currently is being manufactured in cell culture by a German country, and that's going to be, at this point, the target is to make that what we would call the third generation after dry vax and acambus vaccine for use in the U.S. as an attenuated vaccine for use in, for example, immunocompromised people when you needed to vaccinate them against smallpox. Assuming that smallpox could show up again, say as the result of a bioterrorist attack, having this sort of thing is desirable. Of course, whether or not smallpox is going to show up again is debatable.
Yes.
They put this into 3,200 people in Japan.
Mm-hmm.
They seroconverted. They have a good rate of 90% seroconverted. They show some nice lesions here, typical vaccination lesions, and they said the adverse events were quite low.
Mm-hmm.
So that's promising, and I guess they need to go into bigger and bigger populations to see if it really is better. Do we know the origin of dry vax and how it relates to any other known poxvirus strain?
What a great question. The origins of vaccination go back to late 1700s, 1796, where Edward Jenner, an English physician, took material from a milkmaid who had cowpox, which was a common occupational hazard in milkmaids, vaccinated a young boy and conferred in him immunity to smallpox. The interesting thing about that story is prior to that, there were immunization procedures available. There was a procedure called variolation that had been developed, I think, earlier. I forget exactly when. Variolation consisted of taking material from a genuine smallpox case, and scratching it into somebody's arm, and it gave a very nasty reaction. But the case fatality rate from that was less than 1%, whereas the case fatality rate from smallpox was around 25%. So your chances of surviving were increased by at least 25-fold, so this became desirable.
By the way, variolation widespread in China and India by the 11th century.
Okay, great.
Sorry to interrupt.
Oh, that's cool. So Jenner was able to use the same techniques with the cowpox virus, and then actually 6 weeks later challenged the kid with smallpox because variolation was a common practice. He variolated the child and got no reaction, so he knew he had conferred immunity. Now, the assumption has always been that the vaccination virus that we've been using was derived from Jenner's strain. And in fact, Dryvax, I believe, is derived ultimately from the New York Board of Health strain, vaccine strain. If you look in the information about that strain, it's sort of vaguely traceable to Jenner, okay? But there's a lot of, there's a lot of time and missing information there.
Yeah, sure.
The interesting thing is that if you now look at the genome of all of these vaccine strains, which are called Vaccinia, they are different than the viruses that we call cowpox that you can isolate from cows or people that handle them. Mm-hmm. And with a couple of rare exceptions, well, not so rare anymore, that we can talk about, people have been unable to find Vaccinia in nature. So this raises all sorts of speculation about where Vaccinia actually came from, whether that was a virus that was circulating in Jenner's time that subsequently disappeared, or whether it was was originally cowpox and changed through human manipulation in laboratory and clinical situations. Really unclear.
How related is Dryvax to the Vaccinia that you use in your laboratory?
Quite closely.
Closer than to cowpox strains?
Yes, absolutely. So it's a complicated question because it turns out that Dryvax is a very heterogeneous population of viruses. Nobody ever cloned it or anything.
Yeah.
And so when, in fact, they went to make the Acambus strain, what they did was they plaqued out Dryvax, picked a bunch of plaques, and had a look at them. And there are at least dozens of different viruses in Dryvax. Minor variants of one sort or another, but distinguishable, I think, even by plaque morphology and certainly by sequence. The virus that we use in the laboratory is the WR strain for Western Reserve, and that also came from the New York Board of Health strain. So it can be thought of in a way as a plaque isolate from the same seed virus that gave rise to Dryvax and most of the other vaccine strains.
Interesting story there. Well, so basically we can anticipate seeing cell culture grown smallpox vaccines in the next few years being licensed and stockpiled, right?
Absolutely, yes.
These will be more modern, but still derived perhaps by Jenner years ago originally. It's very interesting. It's very, The whole issue of bioterrorism, maybe we'll have time for it later. It's an interesting problem. If not, we'll get you back, talk about it again. So that's good. That was a good paper to get this whole issue. By the way, Rich, there's some people who are not virologists who listen. Just give them a couple-minute primer on smallpox viruses or poxviruses in general. What do they look like and what's the genome and all that? Okay.
Poxviruses are, I think it's important. Smallpox, of course, makes the headlines, but I think it's important to understand that poxviruses are ubiquitous in nature. They infect a wide variety of vertebrates and even invertebrates. For the most part, they are fairly reasonably species specific, so that a virus that grows in one species by and large does not cross to another, but there are exceptions. The cowpox virus infects, actually the cowpox virus is a misnomer. Its natural host is rodents, and the cows are accidental infections from the rodents, and it can be transmitted to people who handle the cattle. Mm-hmm. It's a very unusual beast as viruses go. It's a double-stranded DNA-containing virus with a genome of about 200 kilobases, though if you look at the entire family, there's significant variation. But the Vaccinia virus that we work on has a 200 kilobase genome, so that's enough for about 200 genes. So that's quite large as viruses go, and you'll see why as I go on. It's very unusual structurally. Most viruses have either an icosahedral or a helical nucleocapsid and might or might not have a membrane. Vaccinia is what's called complex, for lack of basically of a better word. It does have a membrane around a brick-shaped particle and then an internal structure that is kind of like a pillow with indentations on each side and some goo between the indentations and the external surface that are called lateral bodies, and presumably the DNA is in the central part of this core. So it's very unusual in structure. It's also unusual in its biology in that it is a DNA-containing virus that replicates in the cell cytoplasm. I'm tempted to say the only DNA-containing virus that replicates in the cell cytoplasm, but that's not entirely true. At any rate, most DNA-containing viruses send their genome to the nucleus where they can take advantage of DNA metabolism and RNA metabolism enzymes that are in the nucleus. Vaccinia has chosen to avoid that bureaucracy and set up shop in the cytoplasm. It pays somewhat of a penalty for that in that it has to encode all of its own nucleic acid metabolism enzymes, which is why it's really of interest to me, because it's almost like a little mini nucleus. It encodes its own multi-subunit RNA polymerase, its own mRNA capping enzyme, its own poly-A polymerase, and most, if not all, of the enzymes involved in DNA replication. So it's a fantastic substrate for looking at basic problems in
Do Mimi viruses
biology.
also carry all of that with them?
I'm not an expert on Mimi viruses, but I would be very surprised if they didn't. And I don't know much about I'm not sure much is known actually about the biology of Mimi viruses. Most of what's known about Mimi viruses is the genomics. But I'd actually have to look at the literature on that and see if they've got all that stuff. But I'd be very surprised if they didn't.
Yeah, Mimis have more. They have all kinds of tRNAs and enzymes involved in translation, and so I would be surprised if they didn't. I think they have a complete topoisomerase system as well, coded in the genome. So, but you're right. Not many people study them in cell culture. They mostly do genomics.
Right.
So what was the one or the exception? You said there might not, Vaccinia or Poxviruses might not be the only viruses that, DNA viruses that multiply in the cytoplasm. What are you thinking of otherwise?
Well, I'm thinking of African swine fever virus, and I'm going to have to update my knowledge on those. But
that's not a pox virus, right?
Well, officially no. Though there's a lot of their biology that they have. Some of the biology and genomics make them look like they're fairly closely related to poxviruses, but they're distantly enough related so they occupy their own family. There's another group of viruses, the Iridoviruses. Mm-hmm. I believe they spend part of their time in the nucleus and part of their time in the cytoplasm.
Now the polyomaviruses, don't they do that as well? A little bit in the nucleus, a little bit in the cytoplasm.
Boy, now you're really testing me. That goes back to the study section that we talked about previously. I don't know. The deal with those is the genomic sequences that you spoke of earlier are replicated out of the genome and assembled into capsids, and in the oviduct of an egg-laying female, and then laid into the parasitized spider. But where the replication must start in the nucleus where the genomic sequences are, but where the actual amplification and capsidation goes on, I don't know.
I found a couple of papers on Poxvirus infections of people. I thought these would be good jumping points for discussion. Two of them are very new. There are reports from Germany and France basically involving people getting infections of cowpox from their pet rats.
It speaks to what you were saying about it being a rodent virus.
Exactly.
Yes.
And in fact, they mentioned that was originally thought that the cow was the reservoir, but it turns out to be rats. Now, correct me if I'm wrong. So the reservoir is the rat or rodents, and then it infects many other mammalian species.
Right. That's my current understanding of the situation with cowpox. Yes.
In this case, they bought a rat at a pet store. In fact, several different people bought rats from the same store. In the German case, 5 people from 2 households near Munich became infected. So presumably the rats from the pet store were contaminated or infected. And then you get lesions on your hand, I presume.
Right. And lots of times these were associated with the rats scratching the people or whatever. So as I said before, the virus does best in a fresh cut in the skin.
So how did it get into the rats to begin with? Like the ones in the store are presumably bred, right?
Well, some of these were imported from another source, but I don't know anything about the source there or the breeding. How they got into the rats? I mean, I have the same question. I'm clueless. I don't understand that at all. I don't understand where they would how they would pick it up. Now, in Europe, another common source of cowpox infection in people is cats.
Right.
And that's there's been a reasonable incidence of that for a long period of time. And I think one of the things that's suggested in these papers that's probably true, although I don't think that there's any good epidemiological evidence for it now, the idea is that some of these diseases may be on the increase because we no longer vaccinate people.
Right.
So we have a poxvirus naive population out there.
So you think the vaccine would confer some immunity to these infections?
I would assume so, yeah. Absolutely.
Now, conversely, would these people get some benefit in the event of a smallpox outbreak, like Jenner's milkmaids?
Absolutely. I would assume so.
Go get a rat.
Go get a rat, yes.
Does this happen in the U.S., Rich?
I've not heard of many cases of this type of thing with cowpox in the U.S. Of course, there was a recent well-publicized outbreak of monkeypox in the U.S. This was, 2 or 3 years ago. There was a similar sort of thing where it's the exotic pet trade. In this particular case, it was prairie dogs that kids had bought from a distributor, though now I'm a little vague on the details, but I don't think the prairie dogs were the primary source. I think they got it from another animal in the pet store from the exotic pet trade. So this was another animal that had been imported from outside the U.S. But at any rate, the monkeypox grows pretty well in prairie dogs, apparently, and some children bought prairie dogs and got monkeypox from the prairie dogs. Fortunately, it was monkeypox. Here's another misnomer. Monkeypox is not monkeypox, okay? Monkeypox has rodents as a primary reservoir as well, but sometimes monkeys pick it up from the rodents and humans pick it up from the monkeys. This is all in Africa, and that's why it's called monkeypox. And there are a couple of different strains of monkeypox around, more or less virulent. Monkeypox is one of the few remaining poxvirus infections that can be fatal in humans. And fortunately, the strain that was imported on these rodents was one of the less virulent strains. And so it was a scare and a nuisance, but nobody was seriously injured.
Are new poxviruses being named by the more standard approach of naming them after the location? Because obviously we've got all these viruses hanging around. You've got cowpox that isn't really in cows, and monkeys that isn't primarily in monkeypox isn't primarily in monkeys. Are newly discovered poxviruses going to follow the more typical nomenclature?
No.
Okay.
I haven't seen any organized effort to catalog and name them in the same way as, for example, the influenza viruses are done. And in fact, in some places, I think the naming is still a little crazy and could use some significant organization. One of the things that we could talk about is the viruses emerging in Brazil, and there's naming issues there as well. But the underlying that is a very interesting story.
Let's do that. Before we go on to those, one more question. These cowpox infections from pets. I assume in general they're not worrisome, except if you are immunocompromised.
Exactly.
Right?
I see it as mostly a nuisance. They can, some of them can turn up some pretty nasty lesions. If you look at some of the published lesions in those papers, it would not be fun to deal with. You can have lesions that are, up to a couple of centimeters in diameter and they itch, and they can be painful, and there can be a lot of swelling. There can be some attendant fever. There can be swelling of local lymph nodes, but by and large they heal completely, and there's no issues aside from the potential for a little bit of scarring at the site of inoculation down the road. So basically, you've just gotten yourself a smallpox vaccination, so the complications, the serious complications from that are all the same as the complications from smallpox vaccination. So you wouldn't want to get an infection from a rat if you were immunocompromised or had eczema. Then you could be in trouble.
So I did find an article about a Vaccinia-like virus infecting humans and cattle in Brazil. So what's that about?
This is a really interesting story because there have been over the past decade or two a resurgence of cowpox disease in Brazil. Once again, I think we have a situation here where when vaccination was widely practiced in the human population, cowpox infections of humans in an agricultural situation was not a problem because the human population was immune. But now that we have a poxvirus naive population, we're seeing a reemergence of cowpox infections in some places, in particular in developing countries. And the farming practices in Brazil are not as developed as in some other countries. This can be more of a problem. One of my closest colleagues is a guy who works in my laboratory, Nissin Musashi, who is actually from Brazil and spent his career in Rio and is retired to Gainesville, of all places, and works with me. I'm very fortunate to have him around. So he's been a big help in understanding this whole story. And in fact, he was a key figure in identifying and characterizing these viruses to start with. At any rate, so there's been an emergence of cowpox disease in Brazil. And by cowpox disease, what I mean is the typical thing where the cows get lesions on their udders, and farm workers, in particular milkers, pick up the disease from the cattle and get lesions on their hands and stuff. And so that can be an agricultural problem and at least a nuisance. My friend Dr. Musashi and his colleagues isolated one of these viruses and characterized it, and found in some of their sequencing that there's a couple of genes that are used to sort of phylogenetically characterize these viruses, in particular the hemagglutination gene, the hemagglutinins gene. And they found first that these things look like Vaccinia-viruses. Now, I've told you that cowpox viruses are distinct from the viruses that we call Vaccinia-viruses in terms of sequence. And so this was a very rare situation where these viruses look like Vaccinia-viruses. Not only that, but this HA gene contained a signature deletion of about 18 base pairs that was not seen in any other vaccine strains around, except Nissin had the brainstorm to look at the sequence of vaccine strains that had been used in Brazil. A lot of countries grew up their own vaccine strains, and this thing was called the IOC strain and was used for the sort of national smallpox vaccination program in Brazil. And lo and behold, the IOC strain has this same signature deletion. And so there is a possibility, and in fact, Nissin believes that the virus that's circulating in Brazil is escaped vaccine that was hanging around in nature for probably decades.
When was the last time it was used in Brazil?
Well, that's a good question. Probably the 60s. All right. Mm-hmm. So it's been hanging around for a couple of several decades in Brazil until the immunity in the population waned to the point where the thing could jump to humans and showed up again.
So where was it hanging around? Some animal that we don't know.
I would assume rodents. of some sort. That would be the most probable reservoir.
And then it would go from rodents to cows, perhaps?
Cows to people. Now, this is anecdotal, but I recall somebody telling me at one time, the rodent to cow transmission may seem like a bit of a mystery. Maybe not. I mean, you can have basically rodents hanging out where the cows hang.
They bite them or scratch them.
Might be one scenario that I've been told of is that when a milking cow lies down in the field, the rodents will actually approach them for a free meal and rub up against their udders and transmit the virus in that fashion.
Sounds like a farm story to me.
Well, maybe one of your listeners will straighten this out.
I'm looking at this paper, and it says the vaccination program took place last in the 70s and early 80s in Brazil. So, been around for a while. That's fascinating.
Yeah. I think that's really a very interesting story.
This paper I have is from 2003. Are there more recent reports of isolation still?
Yes. As a matter of fact, there's a group very active in Brazil that's turning these things up all the time. And there was another recent letter that, let's say, should be appearing in Emerging Infectious Diseases, I think, that talks of this same virus spreading to new northern regions in Brazil. So this is a very active problem that seems to be increasing. It's a good example of genuine emergence and spread of a pathogen.
So it's spreading out of Brazil?
This is not out of Brazil, but into other parts of Brazil. It has the potential to spread out of Brazil.
Has anybody thought of going, or actually gotten out and done this experiment where you'd go out and just start catching wild rodents and looking for poxviruses?
I'm sure, yes. I'm not familiar with that literature, but certainly a lot of that kind of stuff has been done to determine, in fact, what the natural reservoirs are. And that's how people have figured out that rodents are the natural reservoir for quite a few of these agents.
Right.
Yeah, this paper implies that we hardly know what's circulating in the animals, and we need to find out. And this is a recurring question with all virus infections that we talk about now. We need to know what's out there in animals because that's the source of most of them. So it needs to be done. It's hard work. It's hard to get funded because it's not hypothesis driven.
One of my favorite topics. Right. Well, things that weren't hypothesis driven used to be fishing expeditions. But nowadays, if you're cool enough and have serious high throughput science, it's no longer a fishing expedition, it's discovery driven science, and that's okay.
That's right.
Yeah, we didn't know how to pitch it, Rich, so now it's just, deep sequencing and pathogen discovery, right? Which is fine. I
have no problem with it at all. As a matter of fact, my whole life is based on discovery-driven science. So no, it's not. It's not a fishing expedition, Vince. It's discovery-driven science.
Discovery. I have to change my vocabulary.
Also, I'm glad we're getting away from this fishing expedition as a pejorative term because fishing is a good thing.
Yeah, absolutely.
I agree. I don't have a problem with it.
Okay, so I'm going to get on my soapbox here because
I thought you were.
No, not really. You haven't heard anything yet. So I spent my life doing genetics, classic genetic study of poxviruses. So you just isolate mutants and sort them out and then look at them one by one and do infections with a mutant virus and ask what happens and based on what goes wrong, make some deductions about what that gene is normally for. I mean, that's a fishing expedition. But the virus is a lot smarter than I am. Okay, and you give out of that sort of investigation, you learn things that you wouldn't know how to ask about. Okay, so I think there's a real place for fishing expeditions, discovery-driven science. Doesn't all have to be hypothesis-driven.
I agree with you, and I have to tell you, Rich, I'm going back on study section.
Oh yeah, maybe I'll see you there.
I hope so.
I'm thinking that, in particular, if I can get this second grant funded, that I may just volunteer for that job. It would be certainly payback time. And I enjoyed my time on study section with you immensely. That would be a good time.
Yeah, I decided to go back and see what's going on. And I'm going to keep this discovery issue in mind because I think we need to do a lot more discovery.
Absolutely. I agree.
And, I'm a guy, too, classically trained virologist. And I see the value of discovery. In fact, we're trying to do a little for ourselves. And I think the whole issue of viruses out there can teach us so much if we just went out and collected a bunch. And not just in animals, but people too. We have no clue about the steady state level of viruses in the human population.
Right.
We talked about Bob Garcia's study of just polyomaviruses, but those are the ones we know about. What else is out there? Interesting stuff. You had given me a bunch of great stuff to talk about. We hit a lot of them.
I think so, yeah, actually.
But there's one I wanted to talk about, and you mentioned that there are 2 remaining stocks of smallpox that we know about in the U.S. and Russia. And Dick Despommier told me to ask you this question. He wants to know, why is there a vial missing from the Russian stock, and where did it go?
Well, I don't know anything about that, actually. I don't know what the I don't have it. I don't have it. The question always arises out of this discussion of the remaining smallpox strains are the only remaining strains in Russia and the U.S. And what else is that?
We don't know the answer to that, right? No. Yeah, I mean, I think the common understanding is some of it disappeared and we don't know where it is, but it's somewhere. I didn't realize this. You mentioned that WHO is once again considering destroying the remaining stocks.
There are discussions ongoing right now, reevaluating this whole thing, and there is a movement afoot, or at least a discussion of whether or not the remaining stock should be destroyed. And I think that there are a significant number of people who are in favor of doing that.
Let me ask you this, Rich. Let's say they were all destroyed. Having the sequence, would you be able to reconstruct the virus?
Well, there's an interesting question. I believe so, yes. There's only one issue relative to that. You do have the sequence. Technically, it's an interesting problem because poxviruses launch their infection in the cytoplasm, because they do their infection in the cytoplasm, you need the ordinarily when poxviruses infect cells, they bring in with them all of the transcription enzymes that were packaged in the virion, having been made in the previous cycle of infection. And so you can't do a DNA infection. And you can't it would be very difficult to do reverse genetics in the same way as you would do, say, with influenza, where you provide all the replication proteins in trans, because there are so many replication proteins, and the transcription happens ordinarily in this core particle, where from what we know, the structure of the particle itself is very important for getting the early phase of the infection launched properly. So you can't really do the typical sort of reactivation. What you can do is to transfect DNA into a cell that has been infected with another poxvirus. For example, what's typically used is an AV poxvirus, a poxvirus of birds like fowlpox. And the deal there is that the fowlpox replication enzymes, in particular the transcription enzymes, can transcribe the transfected genome and in effect reactivate it and have it replicated and packaged in its own assembly proteins. So it's closely enough related so that there's cross-reactivity among the enzymes, but distantly enough related so that the recombination between the transfecting genome and the infecting helper virus genome is negligible. People have looked for recombination in these sorts of situations and not been able to detect any. So it certainly should be no problem or very little problem from sequence to assemble a genome of a smallpox virus and reactivate it in that way. There's only one remaining issue, and that is that the ends of Poxvirus DNA are really unusual. It's a linear double-stranded genome, but it has covalently closed ends. The DNA is in a continuous loop around the ends, and this has function and implications in the replication of the virus. So think of it as a self-complementary single-stranded circle. And in the very ends, these hairpin ends, there's not complete complementarity, and there's some funny business going on. So those bits at the ends are difficult to sequence. It takes some special technology to sequence them. And although there are sequences available for those ends in some pox viruses, my understanding is that for none of the smallpox strains are there sequences available for the hairpin loops. So my guess is that you could use the hairpin loops from another pox virus and recreate smallpox, but I'm not entirely sure.
Yeah, so it's not a certainty. If it were a certainty, then you could say there's no point in destroying them because we could make the virus anyway.
Absolutely.
So how do you stand on that? Given there is uncertainty, would you destroy them or not?
I'm really conflicted about this. So years ago, I would have said, no, don't destroy them. Because you just never, it's an irrevocable decision. Once you destroy them, you can't have them back. Now, we've just said that you could have them back. And so that dilutes that argument to a significant extent. But I don't like making that sort of irrevocable decision when you really don't know what's going to happen down the road. And when there's no reason necessarily to believe that you can't keep these things appropriately under lock and key. And actually, it's very interesting because the first time that the consideration was being made to get rid of smallpox, to destroy the remaining stocks, was before the whole biodefense thing. And the biodefense thing came up, and sure enough, people dragged out smallpox strains and have been doing experiments at the P4 level on various animal models and cell culture models in the research that has been spawned to look for new poxvirus antivirals and to look for the effectiveness of vaccines and et cetera. So those strains have been useful in all that stuff. Now, the argument can be made now that, well, that's done. And we've done all the research with smallpox that we really need to do. We can recreate the stuff. Let's not keep this stuff around. I don't know. I think that in the end, I would vote against destroying the last stocks for the reasons I had before. I don't see any reason why they can't be kept safely. I hate to make the irrevocable decision to get rid of them. I like viruses. I don't like destroying a species. I don't think it's right.
I think you would agree that we could probably think of more experiments to do with smallpox
Absolutely.
virus. You mentioned before everything's done that we need to do, but next week there might be something else that comes up. So since we probably can reconstitute it, and since there's probably some virus somewhere that we don't know about anyway, it seems to me silly to destroy it. Makes no sense.
That's the way I think about it. Yeah.
I think from a purely scientific view, you would say not to destroy it, and that's the way we should look at this. Don't let the politicians get involved in
this. It's interesting because I think that one of the motivations to do it, I've talked to people who worked on the smallpox eradication campaign. One of my acquaintances in this regard is a guy named Keith Dumbbell, who was very active in the campaign. He was actually in the field. I remember talking to him one time about the difference between different forms of smallpox disease, and he talked to me about diagnosing these by how the lesions felt when you pressed on them. Can you imagine that? I mean, this is a nasty disease, and being out in the field and being presented with people who are sick with this disease and diagnosing it by feeling their lesions. Now, people who were involved in that campaign know from personal experience what a horrific thing this was, and they worked really hard to get rid of it, and they want to finish the job because this is a nasty thing. And I can't help but be somewhat sympathetic with that.
Right. Sure. Yeah, I understand that view, but I think the other views that we've presented are more compelling. So what about you, Alan? You'd keep it or get rid of it?
For pretty much the same reasons that Rich just outlined. If you keeping it as a decision you can reverse. If we decide 10, 15, 20 years from now, well, we really, really aren't going to use this thing. We're really certain we can reconstitute it from the DNA sequence. A future generation can always go and then decide to destroy it. But if you destroy it and you decide 10 years from now, hey, we actually can't reconstitute it from the sequence. We don't have these hairpin ends, or there's some other component that we need, but it's too late. You can't go back on that. And that's a disturbing point to cross, I think.
Let's move on to some email and wrap it up. It was a great discussion. I like that. Before we go on to email, let's have a brief word from the American Society for Microbiology.
On May 17th through the 21st at the Pennsylvania Convention Center in Philadelphia, the American Society for Microbiology will hold its 109th general meeting, the largest annual gathering of microbiologists in the world. Visit the general meeting website at gm.asm.org to view the preliminary program, register for the meeting, or reserve your hotel stay. That's gm.asm.org.
Now, we have lots of reader email, listener email, and it's been accumulating in past weeks because we tend to talk a long time, and I can only read a few. Alan, we may need to have a session where we just read email. Yes. And actually, they're so interesting that we could do that, and it would be interesting. So let me read a couple. And for those who haven't heard their email yet, we'll get to it. I really appreciate your feedback. This one is from Sharon, who writes, "Hello, TWiVers. First of all, I love the podcast and am always anxiously awaiting the newest one. Secondly, I've just listened to the TWiV episode focused on viral evolution and would like to make a brief comment. In defining the viral quasi-species, Dr. Villarreal mentioned that the biological importance of the quasi-species was not fully appreciated until Raul Andino's group showed poliovirus attenuation in PVR mice when infected with poliovirus harboring a high-fidelity RNA-dependent RNA polymerase in 2006. Although the Andino lab did do some outstanding work in this Nature paper, Carla Kierkegaard's lab initially showed these results in 2005 in PLOS pathogens. I often find that they are not granted credit for this discovery, probably since the Andino paper is in Nature, so I thought this would be a good way to spread the word. I'm looking forward to the next interesting discussion." Sharon, yes, you are absolutely right. The Kierkegaard group did publish first on the effect of quasi-species restriction in poliopathogenesis. We'll put a link to Carla's paper, and in fact, it's a great paper. I always use it in my courses, and I'm teaching Tuesday at Rockefeller in a virology course, and we're going to do that paper in class. So I have a feeling you're down in Texas there doing this stuff with Julie Pfeiffer, so I'm sorry, all the Pfeiffer lab, sorry about that. I know because that's Julie's work. We didn't mean to slight it. I actually meant to correct Luis when he said it, but I didn't have a chance. Duncan writes, "Hi fellas, I'm a 31-year-old IT infrastructure engineer living in New Zealand. After years of being an avid armchair scientist, I have decided that I'm going to take the plunge and leave full-time employment to study for a Bachelor of Medical Laboratory Science, specializing in the final year in virology and medical microbiology. My ambition is to be employed in a diagnostic capacity within a medical lab environment. Do you have any words of wisdom for somebody who is passionately interested in microbiology, particularly virology and parasitology, but not looking to work in research? Additionally, I thought I would draw your attention to this site on the chance you haven't seen it. It provides interactive maps of the distribution of various different species of mosquito and the diseases they carry from all over the world. Keep up the good work. Your podcasts are keeping me sane during these last few months of boredom working in the IT industry." I thought the IT industry was the exciting place. I guess not. Anyway, we'll put a link to this mosquito map. It's pretty neat. And as far as your question goes, well, I asked my friend Cliff Mintz, who's an expert on jobs in the biosector, and he said to find some, this summer, a lab where you might be able to volunteer in and work in. And he thought it might be difficult in New Zealand, but that could be the way to go to get some experience. But there's certainly a lot of work for that sort of interest, not research, but microvirology.
Actually, there's a great poxvirus lab in Otago in New Zealand. Andy Mercer runs that lab, and I'm sure he'd love to have a volunteer around.
All right, there you go, Duncan. Bill writes, "Wow, this is by far the most interesting, intelligent blog I have ever come across. Wow. The podcasts are so well done, they are as good as anything I can see on NPR or Bill Moyer's specials." Wow. That's cool. Thank you. I'm an electrical engineer and plan to get my master's degree in biomedical engineering here in Texas. I had intended to go into a track focusing on medical imaging, but the passion I can see in your podcasts regarding the whole spectrum of virology and immunology may convince me otherwise. Since I've been in high school, I've been a huge fan of the icons in immunology and virology, folks like Carl Johnston and the rest of the disease cowboys who stopped the epidemics in faraway places. Too bad many of them have passed along. It would be great for you gentlemen to interview some of these icons. Don't forget about the old school guys at USAMRID, not to mention the older days of Welch and Johns Hopkins. Keep it up. Your podcast is my favorite time during the week. My wife calls me a geek, so be it. My heroes are mine alone, but I don't care. Great email. So, by the way, folks, a geek is someone who is overly obsessed with one or more things, including those of intellectuality, electronics, and of course also virology. So you can be a geek, Bill. I think it'd be great to interview some of the disease cowboys. We'll try and do that. I'm glad you like our podcast, and I'm really glad that it is influencing what you want to do. That's part of the reason we talk about this. We love this stuff, as you can tell, and we want to pass it on to you. Let us move on to our picks of the week. Science Blog of the Week is a site I found called Co-Evolvers. This is by a graduate student at Washington State University, and he writes, "My research interests are focused on co-evolutionary interactions, particularly host-parasite interactions." He's interested in the effect of dispersal or migration on co-evolutionary interactions, local patterns of interaction between host and parasite, and some of the recent posts in his blogs are: why doesn't this pathogen kill me, and why is it taking so long to clear? Parasites can maintain host diversity. Why do I have so many parasites? Evolution of virulence revisited. Pretty neat stuff. A little different from what we usually talk about, although in light of last week's discussion, it's pretty apropos. So check it out. We'll put a link in the show notes. Our science podcast of the week, the Quackcast. And this is his review of quacks, frauds, and charlatans. And he writes, oops, that's not right. That should be supplements, complementary, and alternative medicine, i.e. scam. So it's a very funny podcast where he just trashes alternative medicine. And this is a guy that is not politically correct. He in his podcast once, he referred to the parents who don't get their kids vaccinated as quacks. He puts it right to it. So it's a lot of fun. Check that out. And finally, our science book of the week is called Listen to the Music, The Life of Hilary Koprowski by Roger Vaughan. You may not be interested in Hilary's life, but there's a lot of science in it. You know, this would otherwise be lost. And I have a copy that a former postdoc of mine gave to me years ago. And Hilary was here in April of 2005, and he autographed it. And let me just read a short passage from the first chapter. It starts, chapter 1 is called Jumping the Gun. It is toward the end of a cold January day in 1948 at the huge modern complex that is Letterly Laboratories in Pearl River, New York. And basically, Koprowski had taken a job early in his career at Letterly and was working on developing attenuated strains of polyovirus. And they describe a scene here where they have infected rats with polio, and now they're grinding up the brain and spinal cord in a blender. Koprowski carefully pours 1 cubic centimeter of the cold, grayish suspension into each of 2 small graduated glass beakers. It is an oily glop. Brains are full of fat. The 2 men pick up the beakers and for a brief moment study the contents. Then they look at each other over the rims of the beakers. They drink, tipping their heads back for a long moment until the thick stuff flows sluggishly across their tongues. They make bad faces as they suck the greasy soup out of the corners of their mouths and swallow repeatedly. Koprowski notes that the raw vaccine tastes like cod liver oil. When he can speak, Norton, who is his assistant, asks, "Have another?" "Better not," Koprowski says. "I'm driving."
That's great.
So they actually took the first batches of this attenuated preparation. That's what you did back in the 50s. You drank your own stuff. Sabin did it to his family, too. Of course, nowadays you can't do that. Anyway, it's a great book to learn about how science was done. And Koprowski was a pretty big guy in viruses, so he's still alive. Gentlemen, that was great. I had a really good time talking about pox. Rich, thanks for joining us.
Well, thanks for having me. I've really enjoyed doing this. This is terrific.
Hope you'll come back.
I hope so too. I'm here anytime. And you got a new convert. I guess I'm going to have to just listen every week.
Well, I'm glad you'll come back because we have a good conversation. I always enjoyed talking with you. So,
well, thanks a lot, and thanks for doing this. You know, this is a terrific thing to do.
Thanks a lot. No, I enjoy it. I have a great time, and I learn a lot too. I've just learned a ton. What could be better? Rich, we're going to be doing this live at the ASM meeting in Philadelphia. They're giving us the opportunity to do this in front of an audience, which I think is really great. And that's going to be May 19th, Tuesday, May 19th in Philadelphia. And Alan and I, Dick Despommier, and Raul Andino has agreed to join us. He's going to be at the meeting, so that should be fun. It'll be streamed live on the internet, Rich, so maybe you want to watch it.
Yeah, absolutely.
Watch us in front of a live audience. It should be interesting. All of our listeners, and there are many of them, and I appreciate your listening, go to iTunes and write a review, just a few sentences at the place where you find TWiV. I understand that when you have a bunch of reviews that the podcast will move to the front page of the podcast section on iTunes. And I'd like to be there so we can get even more listeners, because I think virology is great. And the more people that learn about it, the better off we all are. So go write just a brief review, one sentence, I really like this, and so forth. We'd appreciate it. We'll do an experiment, see if that pushes us up. You can follow TWiV on Twitter, PROFVRR, or Alan Dove on Twitter. We love getting your questions. I know we haven't gotten to them all yet. We will, I promise. We love hearing what you want to know about, and we love hearing what we said wrong, so do not fail to correct us. TWiV@twiv.tv. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week for sure. Another TWiV is viral.